Molecular imprinting is a technique for the preparation of synthetic polymers with specific binding sites for a target molecule. This can be achieved if the target is present during the polymerization process, thus acting as a molecular template. Monomers carrying certain functional groups are arranged around the template through non-covalent or covalent interactions. Following polymerization with a high degree of cross-linking, the functional groups become fixed in defined positions by the polymer network. Subsequent removal of the template by solvent extraction or chemical cleavage leaves cavities that are complementary to the template in terms of size, shape and arrangement of the functional groups. These highly specific receptor sites are capable of rebinding the target molecule with high specificity, sometimes comparable to that of antibodies. Molecularly imprinted polymers have therefore been named antibody mimics. It has been shown that they can be substituted for biological receptors in certain formats of immunoassays and biosensors. They are characterized by high stability. Target molecules for which we want to prepare artificial binding sites or molecular imprints, which are normally large complex protein molecules, are identified and selected as template molecules. These template molecules are added to a mixture of monomers and activators and thoroughly mixed. This mixture is allowed to undergo a process of self-assembling, and polymerization, which is actually a guest host, molecular complex, in which the template molecules are trapped in a hardened polymer matrix which act as host. This host guest complex is pulverized, and subjected to a process of solvent extraction, by which soluble template molecules are removed from insoluble polymer matrix. The resultant preparation consists of polymer matrix carrying empty spaces or cavities where the template molecules were originally trapped. These cavities are called molecular imprints, which actually mimic a negative spatial conformation of template molecules. Due to this complementary conformation, these molecular imprints exhibit a special affinity towards original template molecules, and act as artificial binding sites for them. Due to this special affinity, they could be used as substitutes for biological receptors in certain formats of immunoassays and biosensors. Even though molecular imprinting concept is subjected to studies related with modern drug designing, molecular imprinted polymers prepared by this process is difficult to be used as drugs in living organisms, as they are synthetic polymers. Drug potentization in homeopathy has to be studied from molecular imprinting. Since it is totally improbable for even a single drug molecule to remain in a dilution above Avogadro limit, only way the medicinal properties of drug substances could be transferred to and preserved in such a medium is by molecular imprinting. Homeopathy uses water-ethyl alcohol azeotropic mixture as host, in place of polymers, and drug molecules as templates or guests for preparing molecular imprints that could be used as drugs. Since molecular imprints prepared by this process consist of only water and ethyl alcohol molecules, imprinted with three-dimensional properties of drug molecules, they could be safely used as therapeutic agents. According to my view, homeopathic potentization is actually a bio-friendly adaptation of molecular imprinting technology, originally done in polymers. Most important objection raised against this concept is how water can work as a medium for molecular imprinting, whereas water is not a polymer in its classical understanding. But all of us know, water has a lot of anomalous behaviors related with its physical properties such anomalous expansion, viscosity, diffusion, surface tension, melting and boiling points, crystallization, and role in solvation of molecules, ions, membranes, and proteins, which demonstrate water has some polymer-like properties which were not so far sufficiently explained. A recently published research article throws some light into explaining this polymer-like properties of water. This article, titled, "Liquid Water is a Dynamic Polydisperse Branched Polymer," is written by Saber Naserifar, William A. Goddard III, two scientists working at Materials and Process Simulation Center, California Institute of Technology, Pasadena, California 91125. https www.pnas.org DOI 10.1073 PNAS.1817383116 Authors developed a Rex-Pon force field for water based entirely on quantum mechanics. It predicts the properties of water extremely accurately. They observed that strong hydrogen bonds in water connect to form multi-branched polymer chains of 151 H2O per chain at 24.85 Celsius, where branch points have 3 strong hydrogen bonds and termination points have 1 strong hydrogen bond. This dynamic fluctuating branched polymer view of water provides a dramatically modified paradigm for understanding the properties of water. It may explain the angular correlation lengths at 24.85 Celsius, and the critical point at 227 K in supercooled water. This new paradigm for water could have a significant impact on the properties for protein, DNA, and other materials in aqueous media. In supramolecular chemistry, hyperbranched polymers represent highly branched, polydispersed macromolecules with a tree-like topology and a large number of functional groups. During the last 10 years, the rapidly growing interdisciplinary interest in the globular, highly uniform, and monodispersed dendrimers promoted the rediscovery of hyperbranched polymers. The tedious and complex multi-step synthesis of dendrimers results in expensive products with limited use for large-scale industrial applications. In contrast to ice, in which each water makes strong hydrogen bonds to 4 neighbors, researchers showed that upon melting the ice, the number of strong hydrogen bonds drops quickly to 2 in liquid water. These 2 strong hydrogen bonds couple into chains containing around 150 H2O molecules resembling a branched polymer. Authors expect this dynamics branched polymer paradigm may explain many long-standing puzzles of water. Since each H2O on the average makes 2 strong hydrogen bonds, authors next traced out the connections between the strong hydrogen bonds. They found that connecting just the strong hydrogen bonds leads to branched polymer chains with a largest cluster of 151 H2O molecules at 24.85 Celsius containing a main chain of 39 H2O and 15 side chains ranging from 1 to 22 H2Os long. For lower temperatures the largest cluster size increases to 168 at 3.85 Celsius, 177 at 0.35 Celsius, and to 216 at 123.15 Celsius. At a temperature of 24.85 Celsius and below, these clusters may bond to their images in adjacent cells, leading to infinite sizes according to simulations. According to the authors, they have proved that liquid water is a polydispersed dynamic multi-branched polymer in which most H2Os form strong hydrogen bonds to just 2 others, with occasional branch points at waters bonded to 3 H2Os terminating at H2Os. The molecules that are not part of the largest cluster are mainly in small clusters with sizes ranging from 1 to 10 H2Os. At 76.85 Celsius the big polymer chains split into smaller ones, with the number of H2Os bonded to 2 other molecules decreasing. At temperature 123.5 Celsius the connected polymer includes all water molecules, which forms a highly branched polymer. This revelation concerning the polymeric nature of liquid water may have a dramatic impact on our perceptions about water, with possible implications on such physical properties as viscosity, diffusion, and solvation of molecules, ions, membranes, and proteins. To prove that Rex-Pon fully explains the critical point behavior of water requires far more extensive simulations, but these results suggest the potential impact of the paradigm that water is a dynamic polydispersed branched polymer might have on physical and chemical phenomena involving water. We have to explore the dynamics of homeopathic potentization keeping this new paradigm of water in mind. It is not pure water that is used for homeopathic potentization, but an azeotropic mixture of ethanol and water. An azeotropic mixture is a mixture of substances that has the same concentration of vapor and fluid phases. It is basically a mixture that contains 2 or more liquids. A zeotropic mixture basically has constant or the same boiling points, and the mixture's vapor will also have the same composition as the liquid. The azeotrope or constant boiling point mixture is a mixture of two or more liquids whose proportions cannot be altered or changed by simple distillation. This happens because when an azeotrope is boiled, the vapor has same portion of constituents as unboiled mixture. The azeotropic composition of ethanol and water is 95.635 of ethanol and 4.375 of water by volume. Ethanol boils at 78.4 C and water boils at 100 C, but the azeotrope boils at 78.2 C, which is lower than either of its constituents. Perhaps the most important benefit of an azeotrope is the unexpected ability to mix flammable and non-flammable ingredients to produce a stable non-flammable mixture. This is an amazing chemical phenomenon. Azeotropes occur when fraction of the liquids cannot be altered by distillation. Typically, when dealing with mixtures, components can be extracted out of solutions by means of fractional distillation, or essentially repeated distillation in stages. Ethanol and water form an azeotropic mixture at an ethanol molecular percentage of 91 by weight or 96 by volume, which prohibits ethanol from being further purified via distillation. Aqueous solutions at different concentrations in ethanol have been studied both experimentally and theoretically. Azeotropic behavior results from an unexpected concentration dependence of the surface composition. While ethanol strongly dominates the surface and water is almost completely depleted from the surface for most mixing ratios, the different intermolecular bonding patterns of the two components cause water to penetrate to the surface region at high ethanol concentrations in azeotropic ratio. The addition of surface water increases its relative vapor pressure, giving rise to the azeotropic behavior. In water ethanol azeotrope, the water is able to increase the amount of hydrogen bonding between ethanol molecules by increasing the density of hydrogen bond donors and acceptors per unit volume. At a low water concentration, the water also does not interfere greatly with the hydrophobic interactions between the ethyl ends of the molecules. As a result, the average intermolecular interaction for the ethanol molecules is stronger than in pure ethanol, and we have the necessary decrease in the vapor pressure of ethanol. For the water, when it is at low concentration in mixture, each water molecule is surrounded by ethanol molecules, so it has fewer hydrogen bonding interactions than in pure water. As a result, its average intermolecular interactions are weaker than in pure water. When the water concentration gets below what is required for the azeotrope, enough of the alcohol molecules interact only with other alcohol molecules that the deviation from ideality decreases. Considering the polydispersed branched polymer structure of water as well as the peculiar properties of water-ethanol azeotropic mixture, it is not at all irrational or unrealistic to approach the dynamics of homeopathic potentization in terms of molecular imprinting. A lot of studies remain to be conducted on these lines in future. At least, scientific community has to consider homeopathic potentization as a subject of serious further explorations in the field of modern drug designing. Most important primary observation that initiated my logical thought process regarding molecular imprinting involved in homeopathic potentization was that potentized drugs works therapeutically. And the obviously unscientific and spiritualistic explanations given in homeopathy texts for this phenomenon, such as vital force and dynamic energy, was not acceptable for me at all. I wanted a rational explanation for homeopathy that is fitting to the modern scientific knowledge system and its methods. My second observation was that potentized drugs do not work therapeutically, if they are not similar to the given case. Moreover, there are a lot of scientific studies which prove unpotentized ethanol-water mixture in the same ratio of potentized drugs do not have any therapeutic actions. There are scientific studies of eminent researchers showing that potentized forms of a drug can antidote or reverse the biological effects of same drug in their molecular forms, which could be possible only if the potentized forms contain some entities that can act as artificial binding pockets for their molecular forms, which clearly pointed to the possibility of molecular imprints. In vitro and in vivo experiments prove that potentized drugs can antidote the biological effects of their crude forms. This convinced me that the potentized drugs contain some active principles that can act upon biological molecules in a way just opposite to the action of crude drug molecules, which is possible only if molecular imprinting happens during potentization. Then I observed through calculations based on Avogadro constant that there is no chance for any drug molecule to be present in a drug potentized above 12C. Many studies have already proved that potentized drugs and unpotentized ethanol water mixture have similar chemical constitution and chemical properties. This observation indicates that no chemical changes of any sorts happen to ethyl-ethanol water mixture due to the process of potentization. It is a common knowledge that potentized drugs when heated, or subjected to strong electrical or magnetic fields lose their therapeutic properties. This observation indicates that potentization may be involved with some physical changes happening in the ethanol water mixture, that are liable to be reversed by physical forces such as heat, magnetism and electricity. Evaporation rates of potentized drugs and control solutions have been found to differ. It indicates change in hydrogen bond patterns and supramolecular rearrangements. Freezing point of potentized drugs and control solutions are different, which again indicates change in hydrogen bonding patterns and supramolecular reorganization of medium during potentization. In a lot of published spectroscopic studies using different technologies, there are much indications pointing to a supramolecular rearrangement happening during the process of potentization. Spectra were found to be different in potentized drugs and ethanol water control solutions. This showed that ethanol water mixture have undergone some sort of supramolecular clustering and reorganization during potentization. Study of supramolecular structure of water, hydrogen bonding, hydration shells, and supramolecular clusters convinced me that water can exhibit some polymer-like properties at supramolecular level. Scientific studies I discussed in the first part of this presentation has proved water to be a dynamic polydisperse branched polymer, which provides a new paradigm of water entirely different from classical perspective. I happen to notice that ethanol water mixture used in homeopathic potentization is in azeotropic ratios, which led me into a deeper study of azeotropism, and convinced me about the role of this phenomenon in enhancing the polymer-like properties of water. Study of supramolecular properties of ethyl alcohol water azeotropic mixtures convinced me that the hydrogen bond strength of water can be enhanced by the presence of ethyl alcohol molecules in azeotropic proportions, and can restrict the free movements of water molecules, thereby helping in the stabilization of hydration shells. It led me into a deeper study of molecular imprinting, in search of a scientific solution for the riddles involved in homeopathic potentization. A critical study of works done by Benveniste as well as Luc Montaigne regarding what they called memory of water, indicated some structural changes happening in water during successive dilution and succussion. Benveniste and Montaigne might have failed to comprehend the real mechanism involved in the phenomenon of so-called water memory, they observed, which led them to irrational and wrong conclusions. Some Russian scientists have earlier observed a phenomenon they called shape memory property of water, which they could not explain scientifically, since they also did not understand the real process of molecular imprinting involved in it. Study of the phenomenon known as hormesis, which remains still eluding and unexplained scientifically, also led me to relate it with some sort of supramolecular rearrangements happening in water in ultra-dilutions. Observation that potentized drugs act upon biological systems in a way exactly opposite to the original drugs indicated a process of generating three-dimensional nanocavities or intermolecular voids that can act as artificial binding sites for drug molecules and similar pathogenic molecules, which can happen only through molecular imprinting. Then I took up a serious restudy of biochemistry and molecular biology. Study of key lock mechanism, involved in the dynamics of enzyme inhibitions, ligand-receptor, interactions and antibody-antigen, interactions were found to be fitting well to the concept of molecular imprints in potentized drugs. Through these studies, it became more and more clear to me that similia similibus curantur, could be explained in terms of competitive relationships between similar molecules and also phenomenon of molecular mimicry well explained by modern biochemistry.